One of the biggest debates in all of science is whether quantum effects may actually exist in the brain. And over 25 years ago, Max Tegmark famously wrote that the brain is too noisy, too warm, and too wet for quantum effects to be taking place. And so for the last 25 years, essentially, that has been the go-to citation for any dismissal of the potential for quantum effects to be taking place in biology, but especially in the brain. So what's interesting, though, is that there are some hidden assumptions in his original question. And that stems from the fact that he was approaching this problem from the field of quantum computing. And that's because in quantum computing, the bottleneck is indeed how long you can keep qubits in coherent quantum states, because you need a long time to run complex gate operations on these qubits. And you have to bring a pre-existing algorithm to utilize to search with these qubits. And so his question was, well, can the brain maintain coherence in the same way that a quantum computer can? So this is the very subtle switch that took place here, was in the research question that was actually being investigated. What Tegmark was really asking was, can the brain operate like a quantum computer? Which is a much different question than, are there quantum effects in the brain? And his conclusion was, no, it can't operate like a quantum computer. However, he and everyone for the last 25 years interpreted this as there are no quantum effects in the brain. But those are entirely different conclusions, precisely because quantum computers are not brains and brains are not quantum computers for a large variety of reasons. So there are so many differences that were basically unexamined in this search. So sure, it's entirely possible that his conclusion that the brain can't be a quantum computer is true. That's fine. But then the real question becomes are there quantum effects in the brain? And this is where Tegmark and the last 25 years of essentially neuroscience citing Tegmark have entirely missed the point. So the interesting question really becomes then, well, what would it look like to investigate the plausibility of quantum effects in the brain from first principles, basically? Where would you start? And so that's exactly what Jack and I were trying to do in our most recent paper that just got published in Biosystems. And our goal was to approach it from first principles of quantum mechanics and from first principles of engineering and biology. So unlike Tegmark, who started with the assumption that the brain must operate in some way like quantum computers, instead you can ask, well, where in quantum mechanics, at what scale do we know for a fact that quantum effects must or have to exist? And we have an answer to this from quantum mechanics, which means that we can start from one of the most fundamental equations in all of quantum mechanics, which is de Broglie's relation, which says that every particle has a wavelength that is the equivalent of Planck's constant divided by the particle's momentum. And so that means for a particle that the lighter the particle is, or the slower the particle is moving, the longer its wavelength will be. And you can imagine that like why radio waves seep through everything, but gamma rays blast through even the smallest scales. Basically, radio waves are bathing the entire environment because its wavelengths are so long, because they're so slow relative to something like gamma rays, which basically are like a laser and they blast straight through things. So considering that, you can actually make a calculation for the length or the wavelength of a given particle or an electron, for example, at body temperatures at around 310 Kelvin. And when you do the calculation, you get numbers that span in the nanometer range or so, somewhere between 1 and potentially upwards of 4 to 6 nanometers in length. And so immediately we already have a prediction that around those nanometer scales between 1 and let's say 6 nanometers, you should essentially be in the quantum regime, that by default, quantum effects should be happening. And then classical effects would require a significant amount of engineering, a way to cool the system to prevent quantum effects from happening. So then the obvious next question is, well, is there evidence for this? Have we observed quantum effects happening all the time at these nanometer scales? And funny enough is that we actually have hundreds of billions of dollars of evidence of this, thanks to the microprocessor industry and Apple and Intel and company who have literally spent billions of dollars trying to engineer these problems. Because think about what microprocessors are trying to do. They're trying to maintain classical binary computation at the smallest possible scales. And microprocessors now have reached these nanometer scales. So in section 1 of the paper, it goes through a handful of evidence for quantum effects being incredibly difficult to manage at these nanometer scales inside of microprocessors. So they've had to use incredible engineering to create technologies that enable classical computation at these scales. Even the smallest pieces of the microprocessors, somewhere around 3 or 5 nanometers in length, that are actually still functional. So the microprocessor industry is spending billions of dollars to try to eliminate the natural quantum effects that take place at these scales, which generally is quantum tunneling. And tunneling is just a case in which the electrons' orbitals overlap because their wavelengths are larger than the barriers that separate them. And in that case, there's a non-zero chance, thanks to Feynman's path integral, that the electron actually just appears on the other side of the barrier. Because from our perspective, it looks like the electron is traveling through the barrier, but what's really happening is that this computation is taking place outside of space-time, and the electron just appears on the other side of the barrier because the orbital is already there. So then the big question here is, considering that all that we've gathered so far, that de Broglie shows that at these temperatures and these scales, there should be quantum effects. Then the semiconductors show that to maintain classical behavior, you have to spend billions of dollars and have incredibly advanced engineering techniques and cooling techniques to enable microprocessors to continue classical processing at these scales. So then the question is, well, if there's molecular biology at these nanometer scales, which there is, then what is biology doing? Because biology doesn't have the engineering or the hundreds of billions of dollars or the cooling techniques that Apple and Intel would have to maintain classical processing at these sub 5 nanometer scales. So what is biology doing? And we know for a fact that in biology and molecular biology inside the cell, there is tons of molecular systems that are organized at the 1 or 2 nanometer range. And maybe the most well-researched would be in the cytoskeleton with the microtubule tryptophan networks, where they are spaced at 1 to 2 nanometers. And Kalra et al. Has found that when excited, there can be energy migration of up to 6.6 nanometers, which means that these electron orbitals may be spanning upwards of 6 nanometers in length. That's interesting because, the neuronal membrane is only around 4 nanometers thick. So the real question here is, well, then what does biology do? And you can kind of bake it down to almost 2 options. Option 1 is somehow it's classical, but the quantum effects wash out and don't matter, or they're just incidental and errors along the way. Or B, somehow biology has found a way to harness these quantum effects for more efficient energy transfer, for example, or computation. And so that's what we have to get into now, is whether or not there is evidence for one side or the other. So in the 2000s, there was a lot of debate around potential quantum effects in photosynthesis. And there was evidence that was supposedly countering Tegmark's idea that it would be too warm and noisy and wet for long-term coherence to maintain in biology. And this was well-cited evidence, but the debate raged on for essentially 20 years until 2020. And this was the seminal turning point in what has not been fully digested in the biology space as of yet. And this is what Chow et al. Found in 2020. Over 18 researchers from all over the world came together to settle the debate on whether or not quantum effects existed in photosynthesis. And there was really 2 questions they were trying to ask here in this paper. One was, can biology maintain coherence long enough? So this is the Tegmark framing. Can biology maintain coherence long enough for quantum effects to take place? And so part 1 of what they found was essentially Tegmark was correct that biology cannot maintain coherence long enough like quantum computers can to do computation like quantum computers could. But there was a part 2 of their study, and this is what's been completely missed by most people who have read this paper, apparently. So let's just read this together. This is in the conclusion of their paper, and this is the big takeaway. They say nature, rather than trying to avoid dissipation, specifically exploits it together with the engineering of site energies and excitonic coupling to direct energy transport. The role of thermodynamic parameters in driving biological functions is well appreciated on other levels. Here, we see that this principle applies even to the energy transfer processes involved in photosynthesis that occur on the fastest possible timescales. The basic physics behind thermalization is used to impose direction. This simple concept, mastered by nature over all relevant time and spatial dimensions, is truly a marvel of biology. A marvel of biology. And this paper is read as, see, quantum effects can't exist in the brain. What's going on here, man? So this paper has been read as a paper that confirms Tegmark. But what's been completely missed is this conclusion. And the conclusion flips Tegmark's entire thesis on its head. Biology was never in the game of creating quantum computers. Why would it be? It would have to spend tons of energy to keep temperatures near absolute zero or reduce thermal noise. It would be incredibly expensive to pay the cost to consistently maintain coherence for that long. So what instead has biology found? And what this paper says is that the process of decoherence itself is the mechanism in which efficient energy transfer at the fastest possible scales takes place. And they state this really well in another part of the paper. So let's read this here. In simple words, in a world where the behavior of electrons is governed by the fundamental equations of quantum mechanics and that of the nuclei by classical physics, there would be no preferential downhill energy flow, strongly impairing the macroscopic function of these complexes. This relaxation process, by coupling to bath modes, is ultimately responsible for the directed transport of excitation energy, the central function of these light harvesting complexes. In contrast to the population of exciton states, which fail to correctly thermalize, the interexciton coherences can be well described by using a classical description of nuclear motion. So what they're saying is that the process of decoherence is what directs the energy transfer itself. You can think of it like a water cup that has complete water tension. And you can think of the water tension as the quantum state because nothing is happening. It's perfectly stable, for example. But the second that the first drip of water falls off the cup after the water tension breaks, then the energy begins to flow in that way. So then the question here is well, if you can actually pump energy into a system that would otherwise decohere, you could have a bunch of really short quantum states that then decohere. But then that process of decoherence, thanks to the nature of quantum mechanics, Feynman's path integral, energy finds the path of least action, why lightning strikes in the places it does, because energy follows the path of least action. And that's the same way the classical electronics would work, but that is engineered to create the path of least action. But in these systems, in biological systems, the path of least action emerges from the interference of the thermal noise and the energy in the system. And so this is what they're saying is that as long as you can pump energy into the system and maintain a very, very, very fast quantum state, then the efficient energy transfer and computation will happen automatically as the process of decoherence directs the energy transfer. So the decoherence is the mechanism itself for how efficient energy transfer works in photosynthesis. So this is exactly why Tegmark's analysis was so wrong. And Nishiyama et al. In 2024 outlined this as well very explicitly. One of the biggest errors that Tegmark made was assuming that the brain was a closed energy system, completely ignoring the fact that it's a entirely open energy system. And again, in the same way that biology has already found ways to harness these quantum effects for photosynthesis, the most important process for life on this planet, then it's reasonable that if that's happening at the cellular scale already, then couldn't it happen in human biology at the cellular scale in certain scenarios given the right environment? And that seems plausible. And Nishiyama et al. Show why that is. Because again, the energy pumping is what matters. If you provide energy to a system, then you can maintain an excited quantum state for a certain amount of time. And then from there, it'll decohere. But that is how the process works for efficient energy transfer and computation. And so those are major reasons why it's worth exploring whether or not the brain may have quantum effects and whether or not evidence for this already exists. So now we have a lot of pieces we can put together here. We know, thanks to de Broglie's relation, that quantum effects must exist at these nanometer scales. And we know from the semiconductor industry at these scales that to maintain classical computation at these levels, it would require engineering that biology just does not have access to. And then we already have the precedent from photosynthesis that biology already has found ways to harness the quantum effects. And we already know that the brain contains molecular structure at these scales, like, for example, microtubule tryptophan networks at 1 to 2 nanometers of spacing. So then the question becomes is, do we actually see or have potential for quantum effects to show up in the brain at these scales? Is there any evidence that points in this direction? And in the last 5 or so years, there's been massive breakthroughs on exactly this front. So the first big breakthrough happened in 2023 from Kalra et al., who measured electronic energy migration in microtubules with a diffusion length of 6.6 nanometers. And importantly, classical Forster theory, which models energy transfer as dipole-dipole coupling between independent molecules, can't actually account for this diffusion length, because Forster assumes that each molecule acts independently and basically the energy hops one site at a time. But they measured energy migration that exceeds what that mechanism could produce, which essentially definitively meant that something non-classical was going on here inside the microtubules. And then critically, in 2024, Babcock et al. Found an enhanced fluorescent quantum yield across networks of 10 to the 5th transition dipoles. So what they called this was superradiance, that actually there was a collective state when they were excited that acted together and were more stable together than they were separate, which is also something that would not be expected in classical modeling. And then Patwa et al. Built on this and found that quantum yield only drops 3.08% under 5 times room temperature. And so they found that this superradiance was incredibly stable and extremely resistant to high temperatures, which was absolutely unexpected considering Max Tegmark's original hypothesis, because you would absolutely expect that larger systems would lose their quantum properties faster because there are more components and therefore more ways for binding to occur and interference to occur and therefore decoherence to occur. So then the question is, well, what's actually happening there? And something along the lines of the wave functions of adjacent tryptophan networks were overlapping and creating this collective state. And the growing networks actually add more pathways, and these pathways become stronger and more resistant to change thanks to the path integral. Nishiyama et al. Found in 2024, which we discussed earlier, is that these superradiant emissions occur at around 0.2 picoseconds, while the thermal effects actually occur at around 10 picoseconds. So think about that. That's approximately 50 times faster for these superradiant emissions to take place than the thermal effects that would disrupt them. So this creates the entirely plausible mechanism in which Chow et al. In 2020 were describing. This idea that if there is an excited energy state, then it decoheres over time, and that directs the efficient energy transfer and computation of the system. And that's what Nishiyama found as mathematically entirely plausible. So what all of those results show and point to is this idea that the quantum effects are present and organized at this substrate level inside the cytoskeleton. But this paper from Zadeh-Haghighi maybe is my favorite paper because it is showing something fundamentally different, that not only are the quantum effects there, but they may actually change what biology does at a fundamental level. So what they did is they compared different isotopes of magnesium. For example, natural magnesium versus magnesium 25 versus magnesium 26. The only difference here is that magnesium 25 has a non-zero nuclear spin versus magnesium 26, for example, or natural magnesium. So the only difference between magnesium 25 and 26 is literally the nuclear spin. That's a quantum property. And what they found was that under a 3 millitesla field, magnesium 25 actually improved tubulin polymerization while magnesium-26 did nothing. And so why would a nuclear spin change how tubulin organize themselves? This really moves the argument from basically that quantum effects exist at this scale, at nanometer scales, to the idea that quantum effects might be an organizing principle for the molecular structure itself. So now, considering all of the evidence we've gathered from the Broglie to microprocessors to the Chow et al. Decoherence to the quantum biology evidence for microtubules and for energy pumping to maintain enough coherence for efficient energy transfer to take place, and for the spin dynamics that organize tubulin polymerization, you basically get to this point where you can essentially make the claim now, and this is what the paper does in section 1.4, that the brain operates with some amount of quantum effects at the cytoskeletal scale. So if you can confidently claim that the brain does have quantum effects at these scales, which are essentially unavoidable thanks to de Broglie and all that we've investigated so far, the question or the dissenting opinion then becomes, well, so what? Okay, maybe there are quantum effects at the nanometer scale, but they're just going to be averaged out by classical neurodynamics. Like, you don't actually need these quantum effects. They don't actually do anything functional for computation, and especially not for consciousness. So, okay, maybe they exist, but they're completely irrelevant. So we can have the first claim that the brain does have quantum effects at these scales, but it doesn't necessarily mean that they matter yet. So now we have to investigate, well, is there evidence that connects these quantum properties, these quantum processes that we've now outlined at these nanometer scales, to higher order cognition or to consciousness itself? So considering this, anesthetics actually provide the best case scenario for testing this, because anesthetics disrupt essentially everything. You can start from there and start to narrow in on where exactly these quantum effects may be coming into play for consciousness, for example. And if microtubules are heavily involved in higher order processes and these quantum processes are unavoidable at these scales, then you would expect that a stable microtubule would be very important to computation and to consciousness, for example. And you would expect that if it's destabilized, it would disable consciousness or reduce the effectiveness of your cognition or in consciousness in general. So there's a lot of predictions you can make just based off of this alone. So the first question is, well, do anesthetics, perturb the microtubule or the tubulin at all? And that's exactly what Kalra was searching for in their 2023 study. So they found that isoflurane reduces the diffusion length from 6.6 nanometers to about 5.6 nanometers or so. So between a 12 to 15% reduction thanks to anesthesia. And so you're already seeing this delocalized effect from anesthesia alone on an isolated microtubule. So then the question becomes, well, yeah, okay, microtubules are affected by anesthesia, but so is everything, because anesthesia binds to essentially everything. So then the question becomes is whether this particular disruption of anesthesia has any significant importance and tracks one of the longest standing puzzles in anesthesiology, which is the Meyer-Overton correlation. So they found that the potency of anesthesia, which is basically how much you need to knock you out, essentially perfectly tracks with the solubility of the thing you're injecting it into. And so for over 100 years, no one had found a correlation that actually mapped this, or something that actually would explain why this was the case. What Craddock et al. In 2017 found was that they modeled anesthetic interactions with tubulin, and they found that, for example, like tryptophan exhibited this collective oscillatory modes between 480 and 700 terahertz. And anesthetics actually shifted these frequencies, and certain terahertz correlated with the clinical values of an R squared of 0.999. And so this was the first time that a single mechanistic variable at a specific target reproduced Meyer-Overton at this type of fidelity. And so there hasn't been another molecular target that has come close to matching this. So even with this like perfect Meyer-Overton correlation to microtubules and to tryptophan, a skeptic could still argue like, well, yeah, that's nice and all, but you'd have to test everything before you can conclude that microtubules are the only one that can do this. So that's not really feasible. But is there some other way that we can actually narrow this down? And maybe you have to start off first with, well, what else that can be affected in this way? And then what else can be constrained in this way? And that's where we can go from here. But this is actually very interesting because the beauty of anesthesia is that we know for a fact that it works on all living organisms. This has been known since the 1800s, and these experiments have been done even all the way down to single-celled organisms. For example, in 1974, None et al. Found that Tetrahymena, which is a single-celled organism, there's no nervous system or anything, that it loses its motility and then recovers with the effects of anesthesia. And so you can narrow down the target to something that must be in all eukaryotic cells. And that really rules out the neuronal level specific mechanisms. So Mike Wiest in 2025, who I highly recommend you checking out his work and his papers. We'll talk more about his work in his lab here soon. But he wrote about exactly why this is so critical because, for example, most theories of anesthesia involve like ion channels or higher order neuronal level effects. But what he's pointing out here is that, well, if MAC, like this concentration level, varies little across the species, but ion channels and neuronal effects vary drastically across species, well, then if anesthesia is working even on single-celled organisms and at the most simple level of biology, then essentially all of these higher order theories of consciousness are ruled out. And something inside of the cells has to be responsible or critical, at least, very important. It may not be the only thing, at least, but it has to be at least very critical to the conscious experience itself. And so this is why he identifies that it has to be something inside the cells. And we know that microtubules are inside all cells. And in the brain, there's even a higher concentration of microtubules in neuronal cells. And so within all currently identified candidates, the microtubule cytoskeleton is the conserved substrate that satisfies all of these constraints simultaneously. The dissenting opinion could still say, okay, sure, whatever. There are quantum effects inside the cell. Sure, there might be quantum effects in the microtubule. Sure, they might also be ruined by anesthesia. And sure, maybe they play some type of role in consciousness, but it's not actually that important to the maintenance of consciousness. And the consciousness that we care about as humans and as for animals isn't actually involved at the cytoskeletal level. And so then the question is, well, what would happen if you could protect microtubules? Sure, there might be other aspects of the cell that are important to the processes of consciousness, but could you start isolating these parts of the cell, these molecular structures, by protecting certain parts of them and then injecting anesthesia and seeing what would happen? And that's exactly what Mike Wiest's lab did in 2024. So his lab and Kahn et al. In 2024 gave rats a microtubule stabilizing drug, epothilone B, before exposing them to isoflurane or anesthesia. That's because epothilone B binds to microtubules, specifically to tubulin, and prevents depolymerization of the microtubule lattice. So this has been used for fighting degenerative mental diseases or like Alzheimer's, for example, and for fighting cancer. And the result was extremely significant. The rats took significantly longer to lose consciousness with a Cohen's d size of 1.9. And so if microtubules were incidental to consciousness and they just weren't that important, then protecting them shouldn't have any effect on when consciousness is lost, except it has a massive effect. And you et al. Just recently in 2026 built on this as well and extended this to the same test to Tetrahymena, which is this single-celled organism that we just talked about. And isoflurane reversibly slows swimming. And so when you give them anesthesia, they slow down and they come to a screeching halt, essentially. But then if you actually incubate them in epothilone B before giving them the isoflurane, there's a pretty significant improvement in resistance to this slowing. So then they separated it, and you can give these Tetrahymena epothilone B, and that does nothing to them. So there's no effect on reducing their swimming. But then if you give them epothilone B, and then you give them isoflurane or anesthesia, you actually get to see that, again, the epothilone B seems to reduce the effect of anesthesia even on a single-celled organism. So when you combine these, what are we seeing? We're seeing that the same compound that delays unconsciousness in rats and mammals also gives anesthetic resistance to single-celled organisms. That points to this idea that the mechanism must operate through a property of microtubules that is conserved across eukaryotic life independently of anything neural, or at the neuronal level, at least. And so there's actually a really important subtlety here when you put this in the context of the Kalra study from 2023, who found that when you give anesthetics to microtubules, it reduces their energy migration by 12 to 15%. So that's reducing the quantum effects. But that's important because the electrostatic environment of the microtubules, of the tubulin surface specifically, governs both classical processes of energy transport and what we're seeing here of quantum properties of energy transport. And so you have both going on simultaneously, and epothilone B protects both of them. And so in that regard, it's no surprise that when you inject anesthesia, it reduces the quantum properties, but the classical properties remain, which would be potentially why you can remain alive, but not conscious. And so there's a separation here where we're seeing that the classical effects seem to be intact, but the quantum effects seem to be lost. And then in the same way, you still live when you're under anesthesia, but you're not conscious. So this points to the idea that the quantum effects are directly related to the conscious state themselves. So let's consider where we're at right now and where this dissenting opinion would potentially have to be. So maybe the dissenting opinion may say something like, okay, so even if all of this is true, even if anesthetics disrupt the quantum variable, even if potency tracks it, even if the target is conserved across all eukaryotic life, even if protecting microtubules delays unconscious, maybe all of that could work in a controlled experiment, but still be irrelevant to what happens in a living brain. So this is basically Tegmark's argument coming back full circle here, is that, well, sure, maybe all of this is true, but at the end of the day, it's not going to happen in the brain. And so this is why this argument is completely missing the point here. And that's what the first section of this paper had to establish first, is that it's already happening in the brain. And so once you get to the point of accepting the premises that quantum effects are here already, that they happen at the nanometer scales, you can't ignore them. And that's why this whole chain of evidence works its way up to the point that consciousness itself from the anesthesia evidence seems to point to the fact that microtubules and the quantum effects in microtubules are absolutely critical to consciousness itself. And this is exactly why Tegmark had completely missed the point, and completely missed the point from a biological perspective that quantum states can be maintained as long as energy pumping occurs. Because the biological system, the brain, is an open energy system, unlike quantum computers that have to be a closed energy system. Just like a laser versus a lamp. In a laser, as long as you're pumping energy into the laser, it has a really, really powerful quantum effect essentially that creates the laser. Well, if you have a very small amount of energy, you get a lamp. And so the energy pumping is actually part of the mechanism itself that enables quantum states to be functional. And this is exactly what Haghighi et al. In 2023 calculated. They calculated that action potential energy pumping delivers between 5 and 50 kBTs per second to membrane dipoles. And the minimum threshold for sustaining coherent dynamics is about 0.3 kBT per second. So the brain can exceed this minimum threshold by roughly 100 times. And so the coherent energy delivery is not the problem here when it comes to how quantum states can propagate outwards and have effects at the cytoskeleton level. And that's where the Nishiyama paper comes back into play here, is that as long as this energy pumping from action potentials is being delivered to the cytoskeleton level at a rate that fulfills this threshold, then we already know mathematically that these operations can occur about 50 times per cycle before thermal interference decoheres the states. And this allows for the efficient energy transfer from the quantum mechanical perspective that we identified earlier on. The key to maintaining the effect of quantum states at the cytoskeletal level, and therefore potentially the effect that it has for consciousness and for higher order cognition would be the energy delivery to these levels. And so it's no surprise that anesthesia has such an important effect, because what Weininger and McIver found in 2006, and there's a bunch of support on this front for the effect that anesthesia has on energy migration, even classically, they found that clinical isoflurane depresses synaptic output by roughly 60%, while basic spike conduction persists. What we're getting to here is that anesthesia operates basically at this dual impairment level, and that at the molecular substrate, it's impairing the microtubules, for example, like with what Kalra found with energy migration, with like up to a 15% reduction or more. And maybe even more importantly, the anesthesia is massively reducing the energy supply to these levels. So not only is it weakening the molecular substrate, but it's reducing by massive degrees the energy supply to the cytoskeleton. This would be the threshold failure that Cavaglia et al. Were finding there, is that at 0.3 kBt per second, if you go below that threshold, processes just stop. And that would point to the reason why anesthesia works so fast and so effectively to put people unconscious, while classical processing remains and we can all stay alive while under anesthesia. So now we're getting to the climax here of the argument. So let's take a step back and really look at what we've established here. What we've established in the last few sections is that anesthesia works on single-celled organisms with no neurons. And 2, protecting microtubules delays the effects of anesthesia in both rats and the single-cell organisms. Then 3, the energy supply to the cytoskeletal substrate is what maintains these states. And there's a threshold at which this supply is enough or not enough. So all of this is pointing to the idea here that higher order cognitive function, neural networks, the synaptic processing, those things at the synaptic level, all of that is not fully responsible, at least, for whatever anesthesia is eliminating and this loss of consciousness. And so there seems to be 2 aspects here, which is kind of this higher order cognitive function, for example, our sense of self and higher order cognitive metacognitive thinking, and this substrate level functioning, which is what's happening in the cytoskeleton and the energy processing and the informational processing that must be occurring at the cytoskeletal or inside the cell, at least at that level, the nanometer scale level. So what's interesting, though, is that the anesthesia eliminates both. It eliminates both the higher order thinking, the metacognitive thinking, the sense of self, and it eliminates and inhibits the substrate level, the cytoskeletal level, the microtubule level, this level of processing at the nanometer scales. So then the interesting question becomes, if anesthesia eliminates both modes, this lower level, substrate level, and the higher order thinking, is it possible just to eliminate one of the modes? And if so, what would that look like? Specifically, could we eliminate the higher order thinking and isolate the substrate level, since that's where the evidence is pointing to for these quantum effects to be so critical? Because theoretically, someone could still try to argue that, okay, maybe the quantum effects matter to some degree, but they're not really that critical. They're just sort of like the foundation of which consciousness is built on. Maybe consciousness is a little bit more complex than I was originally giving it credit for. And so maybe perhaps consciousness is a mix between the higher order and the lower level, but the lower level isn't really that important for consciousness. So that's why, if there's a way to eliminate the higher order thinking while maintaining the substrate level thinking, you can then eliminate that dissenting opinion and isolate consciousness at the lower levels. And this is also interesting to think about in the context of Jack and I's other paper, which points to this switching mechanism between modes of thinking. There's a fast mode of thinking and a slow mode of thinking, or potentially this quantum substrate mode of thinking and this neuronal mode of thinking. And the 3 dials that the evidence was pointing to was, 1, the energy supply. Where is energy flowing? And this goes back to our idea about the threshold at the cytoskeletal level. And then the integrity of the cytoskeletal level. So this is, again, pointing to the idea that Kalra measured, that anesthesia reduces the structural integrity of microtubules themselves. And then 3, is the electrostatic environment, how noisy it is in the environment, whether that's electromagnetic noise or thermal noise or something in between. Those are the three dials that are most important to what mode of thinking you may be in here. So we know that anesthesia impairs the first two, the energy supply and the structural integrity, and then it shuts down the electrostatic environment because everything goes unconscious when anesthesia is administered. So what would the opposite look like? Could you enhance the microtubule substrate while eliminating the electromagnetic environment? And then that would help isolate the effects themselves. And this becomes even more important because at the neuronal level, we know that the default mode network, thanks to Carhart-Harris's research, some parts of the default mode network use up 40% of the energy above the brain's baseline. So it's an extremely expensive process to maintain the higher order cognitive functioning. So if you can turn off the higher order cognitive functioning, then what happens? And the beautiful thing about this is that in the last 15 years, there's now extensive evidence on exactly this front. And this is where I think the paper really solidifies its argument, because psychedelics do exactly this. It eliminates the higher order thinking and completely flattens the electromagnetic environment of the default mode network and higher order cognition, while simultaneously preserving the underlying substrate and not affecting it in any significant degree. So then we can look into the evidence here about what's going on when psychedelics are administered. Evidence for this is extremely strong recently. Timmerman et al. In 2025 found with participants who were administered 5-MeO-DMT would experience this radical disruption of self with a preserved waking awareness. So they would be there, but there would be no one there to experience. Their sense of self was gone, but this experience of presence would still exist. And interestingly enough, is that they found is that the experience would actually intensify, that the sense of being and presence was stronger when administered with DMT, even though there was no self there to experience it. And interestingly enough, what they found was in EEG is that the alpha reduction was absolutely flatlined. And while other electromagnetic bands weren't nearly as affected, delta and theta, there was very little change. And so from this open system perspective that we are talking about, this whole mechanism, that's exactly what you would predict, because the energy supply to the substrate would be maintained and actually improved as the default mode network shuts down. So you'd have more energy supplied available to the substrates to maintain coherent states at those levels. So broadly, the fact that awareness actually deepens or intensifies when self-referential processing and the alpha bands associated with the default mode network go quiet point towards the subneuronal, the intracellular, the cytoskeletal effects being absolutely critical to the maintenance of conscious states. So what Timmerman is establishing is that awareness intensifies when the DMN shuts down and self-referential processing is removed. Of course, then someone could still say, well, that doesn't actually mean anything for microtubules. But that's where the Zadeh-Haghighi finding in 2023 becomes so important, because they were also testing for psychedelic compounds and how they relate to microtubule stabilization. And what they found is that psychedelic phenethylamines directly enhance microtubule polymerization at the substrate level. So in the same way that anesthesia was disrupting microtubules and reducing their energy migration by 12 to 15%, as Kalra et al. Found, Zadeh-Haghighi is finding that certain strands of psychedelics were actually enhancing microtubule polymerization. And this allows you to create the hypothesis that it's highly likely that in essentially all psychedelics, it doesn't disrupt the microtubule substrate. And in some cases, it might even actually enhance the microtubule substrate. Now, think about what we've established here in the last few sections. Anesthesia impairs both levels simultaneously, the substrate cytoskeletal level, even up down to microtubules and tubulin polymerization and energy migration in tubulin. And at the same time, it impairs the higher levels. You don't have higher order thinking when you're unconscious, for example. And so what we can assume is that during anesthesia, the energy supply drops below the threshold that Cavagli et al. Determined, and everything goes down, consciousness disappears. Psychedelics allow you to isolate the variables, because you administer 5-MeO-DMT, it binds to the 5-HT2A receptors, and then you eliminate higher order cognition, but conscious awareness persists and actually is enhanced, which then you can eliminate the idea that the aspects of conscious awareness are associated with the default mode network and alpha bands, for example. So you're actually isolating more variables here again. Action potentials are still firing, so you're still having the energy supply down to the cytoskeletal substrate. DMT and psychedelics aren't disrupting the same things that anesthesia is disrupting. So now you're isolating variables to a way higher degree. And then the Zadeh-Haghighi finding shows that actually these compounds, psychedelic compounds, can actually enhance microtubules, which would also be expected considering the phenomenological reports that perception and awareness can actually be enhanced when self-referential processing and the default mode network are entirely shut off, higher order cognition entirely shut off during psychedelic experiences. We have finally arrived at the full chain. Why Max Tegmark is entirely wrong and asking the entirely wrong question about quantum processes in the brain. And it starts with the fact that de Broglie already shows us that at nanometer scales between 1 and, let's say, 10 nanometers, there you are expected to have quantum effects at those scales. And semiconductors confirm exactly this, that at these scales, to maintain classical computation at scales in which you would expect quantum effects to exist, you have to insert tons of energy, tons of extremely complex engineering that biology has no access to maintain classical processing at the 1, 2, 3, or 4 nanometer scales. And then we know for the fact that the brain does indeed operate at these scales and has plenty of molecular structure at the 1 or 2 nanometer scales. And then Chow et al. Showed us that biology harnesses quantum effects at these scales through the process of decoherence itself. That decoherence itself is the mechanism in which efficient quantum energy transfer takes place inside of biology for photosynthesis at the cellular level. Microtubules exhibit multiple quantum phenomena at these scales, including energy migration at 6.6 nanometers, superradiance, and that these quantum superradiant effects are extremely robust even at 5 times room temperature. And importantly, we found that Max Tegmark completely missed the fact that the brain and biology is an open energy system, while he was coming from a quantum computing closed energy system perspective, and that energy pumping is actually how these quantum states exist in biology itself. And all you need is the energy pumping to exist at a rate faster than thermal decoherence. And the math works that you can actually have 50 of these excited quantum states in the time that one cycle of thermal decoherence can take place. So there are definitively quantum effects in the brain. So the question is, do these actually matter for consciousness or higher order cognition in any sense? And that's where all of the anesthesia and psychedelics evidence comes into play. That anesthesia eliminates both modes. It eliminates the substrate mode and even down to single-celled organisms, and it eliminates higher ordered thinking, and it has an effect across all of life itself. And anesthesia potency inside microtubules tracks the Meyer-Overton correlation to 0.999 R squared. And then importantly, protecting microtubules actually preserves or delays unconsciousness when anesthesia is administered. And then in the opposite effect, when you administer psychedelics, for example, you eliminate the higher order cognition, but all of the substrate, molecular, cytoskeletal cognition is entirely intact. So all of this points to the idea here that Max Tegmark was entirely wrong about quantum effects in the brain. That quantum effects don't only exist in the brain, but they're absolutely critical to the maintenance of conscious states and likely very critical in the processing for even higher order cognition at a significant level. And both likely interchange together, and the higher order works with the lower order. And that's what Jack and I's additional papers show, is that there's plenty of evidence for how the substrate communicates with the neuronal scales, and both work together to organize how we perceive the world on a moment-to-moment basis. So hopefully you enjoyed it, the paper will be linked below. The book will also be linked below, which expands, on a more theoretical basis on the ontological implications of a lot of these findings. And our other paper, the switching mechanism paper, will also be linked below. So thanks for hanging out with me, and hopefully you enjoyed this absolute exploration into the brain, into quantum effects into the brain. And this has massive implications for what the future of quantum biology itself, neuroscience, and consciousness studies could look like, and how we understand how the brain works, how humans work, how we create meaning in this world, even where consciousness began, and how humans can interact with each other considering all of these things. So hopefully, we definitely have a lot more to talk about here on the channel, and a lot more research is coming soon on that front as well. So thanks for hanging. We'll talk very soon. Bye.